Good afternoon, everyone. Can you all hear me? Great. So my name is Sanchutha. I'm a microbiome scientist at the Medicines and Healthcare Products Regulatory Agency. I'd like to thank the Magnus Group for the opportunity to present a snippet of our work today on the development of reference reagents for antimicrobial resistance, gene detection, and surveillance. So just a few words on who we are. We are a World Health Organization international laboratory for biological standardization. So we develop and manufacture more than 90% of WHO international standards. We exist to ensure the safety of medicines, medical devices, and blood transfusion products for the UK. I'm part of the microbiome group, so we are dedicated to regulating and standardizing microbiome science and research related to diagnostics, surveillance, and its role in medicinal products and therapeutics. And we lead the microbiome standardization program and have established the first 3 WHO international reference reagents for microbiome research. So a lot of our work also involves applying our established approaches to antimicrobial resistance detection and surveillance, which has never been more important today, given that it is a declared global health threat. In 2019 alone, 4.95 million deaths were attributed to bacterial AMR, and this number is forecasted to rise to 10 million deaths per year by 2050. AMR is not region or country specific. No country is protected from antimicrobial resistance. And there are global and national action plans that have been designed to address specific aspects of antimicrobial resistance. And this includes reducing unintentional antimicrobial exposure, optimizing the use of antimicrobials, investment in innovation, equity, so ensuring that everyone, regardless of location and socioeconomic status, has access to the necessary diagnostics and antimicrobials, and also being a good global partner. So AMR is a global problem, and that involves sharing knowledge, data, and technology. However, there are some serious gaps with sensitive, accurate, and synchronized national and global detection and surveillance efforts, which are crucial for providing that data that will help to determine how to address this threat. So there are several approaches that you can use for AMR characterization. These tend to be grouped as phenotypic and genotypic approaches. Clinical settings, certainly back in the UK and in many other countries, remain quite reliant on phenotypic traditional approaches like distribution sensitivity testing, variations in the minimum inhibitory concentration assay, and also in mass spectrometry, so the detection of enzymes and proteins specific to particular AMR mechanisms. We also have genotypic approaches like PCR and DNA microarrays for the detection of specific genes related to AMR. Sequencing appears to be the only option that provides a holistic view of microbial AMR gene burden in a biological sample or in a natural environment. So it's a rapid and versatile approach that can be used for AMR gene detection, and its benefits were clearly observed with the previous pandemic when we used metagenomic sequencing for wastewater surveillance, COVID, SARS coronavirus 2 detection. And this was done in countries all across the world, even in countries with limited resources. So imagine if we can do the same for AMR gene detection. And this approach could be used for AMR surveillance of wastewater, agricultural sites, environmental deposits, and patient samples. And it would enhance our understanding of how AMR spreads, leading to better stewardship. So before we can make this happen, we have to ask ourselves some important questions, like what methods we should use. So metagenomic sequencing workflow for AMR gene detection has several stages, and each stage can be done using a variety of different approaches. And this is particularly relevant to DNA extraction, sequencing, and analysis. So the second question is, what information will this data provide, and what is its biological relevance? So we learn about the AMR potential of a microbiome for an environment or a population. We learn about the mechanisms of AMR spread, the advantages that it may hold over other methods, including more traditional approaches, how we can use it in a clinical context, but also recognize the challenges that may come with using this approach and the limitations at present. So, for example, we recognize that AMR is not just about resistance gene detection. There are more complex layers of phenotypic expression of resistance that need to be profiled alongside the resistome. However, given that there are no standardization approaches for AMR, a good definitive starting point that we could consider is the resistance gene detection, presence and absence of resistance genes. And then once we've laid that foundation, we can then think about building on it with profiling additional layers of resistance. So the third point is, what would be the impact of this data on public health? We'd attain a better understanding of AMR, of the AMR landscape and how it spreads. And this information could feed into better prediction, control, and preventative measures for future outbreaks, as well as the development of more targeted therapeutics. And lastly, how can we trust the data that we get? So what measures can we implement to ensure that we are attaining a true representation of AMR? So the microbiome group at the MHRA have dedicated a lot of effort into instilling trust into metagenomic data. And this involves profiling the differences in methodologies used in DNA extraction, sequencing, and analysis from laboratories across the world. And it also involved the development of WHO international reference reagents for microbiome studies. So what we hope to do is apply our knowledge and this framework to the standardization of AMR gene detection using metagenomic sequencing. So we decided to carry out a pilot study for AMR gene detection to see what we would anticipate. And this involved analyzing the same sequencing data using 5 different analytical pipelines. So the sequencing data was for 20 bacterial strains associated with gut microbiome health, and also the 2 DNA microbiome reference reagents that we have developed. And the 2 DNA microbiome reference reagents were composed of the same 20 bacterial strains from the gut. The difference being that one reference reagent contained equal DNA concentrations of all 20 strains, and then the other contained staggered, uneven concentrations of the 20 strains. So the green boxes represent different bioinformatic pipelines that we use to analyze the 20 strains and the 2 DNA reference reagents on the x-axis, and the y-axis shows the number of genes that were detected. Each color represents a particular resistance to a certain antibiotic. So the general point here is that you can see that there is a clear variation in the number of resistances detected across the 5 different analytical pipelines. So to give you a more specific example, if you take the individual bacterial strain that was sequenced using the 5 different analytical pipelines, if you take the first strain, 10 resistances were detected with pipeline 1, 10 resistances were also detected with pipeline 2, and then half as many resistances were detected with pipeline 3, and then no resistances were detected in pipeline 4, whereas 27 resistances were detected with pipeline 5. So at present, I could not tell you which pipeline provides the truth about antimicrobial resistance, which is alarming given the implications that this would have on all AMR data that's generated from metagenomic sequencing. So this is the problem that we're trying to address, and this is why it's important to standardize AMR gene detection by metagenomic sequencing. So aside from providing the true representation of the resistome, standardization would enable data reproducibility and comparability, and it would facilitate national and global harmonization of that data. It would identify limitations and strengths of the sequencing and analytical methodologies that are being used. It would strengthen the reliability of this data from diagnostic and surveillance efforts. It would ultimately bring confidence to all of this data. So we're at present developing 3 antimicrobial resistance detection reference reagents. 2 of them will be DNA-based and 1 will be whole cell-based. The reference reagents will be proposed as WHO international standards for the absence and presence of AMR genes, and we will thoroughly characterize these reagents to determine the true AMR gene content. And these will help AMR stakeholders decide on the best DNA extraction, sequencing, and analytical methods used for their own gene studies. More specifically, the reference reagents will be used to profile how the methods handle single strains, but also more complex polymicrobial samples, and that's where the reference reagents come in. And ultimately, the goal is that these reference reagents will harmonize sequencing and analysis for AMR gene detection across the world. So we use 7 bacterial strains from the escape pathogen spectrum to make up our 3 different reference reagents for AMR gene detection. The first 2 are DNA-based, and the first one is present with all bacterial strains being present in equal DNA quantities. The second has the different strains present in uneven or staggered DNA concentrations. And the whole cell reference reagent is essentially the bacteria present as fixed cells in equal cellular quantities. So we used different sequencing approaches and different analytical pipelines to test the AMR gene detection reference reagents. And the questions that we were asking were, are we able to identify all the AMR genes present in the heterogeneous samples? Are these reference reagents able to assist with choosing appropriate sequencing and analytical methods for AMR gene detection in microbiome studies? And with a global collaborative study, we hope to determine whether they contribute towards harmonization of sequencing and analytical methodologies used, as well as in data surveillance. And do they facilitate the comparability of data sets between the different laboratories across the world? So this is a summarized flow diagram of what was involved to develop these AMR DNA standards. So very briefly, we grew the bacterial strains, the DNA was extracted, and their identities were confirmed and verified. And then we combined the DNA at equal concentrations and also at staggered concentrations. And then once the standards were made by the standardization team, we then analyzed them ourselves to confirm the relative abundances and the vial consistency. The whole cell standard involved a similar process with some key differences. So once the bacteria were grown, they were fixed and then plated, and they were incubated for a 28-day incubation period to ensure that they were properly fixed. And once their identity was confirmed, we then verified the cell numbers by FACS and then combined them in equal cellular quantities. And once the standards were made, we then analyzed them for their relative abundance and vial consistency. So these were the different sequencing methodologies and analytical tools that we used to analyze the data. So when we say analyze the data, we analyze the 7 individual bacterial strains as well as the combined reference reagents as well. In total, there were 30 different combinations that were used to analyze this data. So these are some preliminary findings, what we have so far. The sequencing data that we have got for the majority of the study is currently being analyzed. But to give you a snippet, this is an individual strain that was analyzed by the different analytical pipelines. And then the number of AMR genes detected is on the y-axis. The different colored columns represent a different sequencing methodology that was used. So there were 2 main points to this, which was that the number of genes detected seemed to be pretty consistent, reasonably consistent along the different sequencing methodologies used, but there was an evident variation in the number of AMR genes detected based on the analytical approach used. And this varied between 8 and 294 resistances. In the case of our DNA-based reference reagent containing an equal mix of all the bacterial strains, a similar thing was seen. So the number of AMR genes detected was pretty consistent along the sequencing methodologies. However, there was a clear variation in the number of resistance genes detected when we varied the analytical pipeline that was being used. And in this case, it varied somewhere between 57 and 417 AMR genes. So once we've developed these reference reagents and they've been vetted from our side, they'll then be evaluated in a global collaborative study before they are established. So previously, we carried out 2 global collaborative studies for our microbiome reference reagents, and these essentially involved handing over these reference reagents to participant laboratories across the world so that they can then be analyzed. And we hope to apply the same framework to the standardization of AMR reference reagents. So with the first global collaborative study for our DNA microbiome reference reagents, 23 participant laboratories from 11 countries were involved in analyzing those reference reagents. And in our second WHO collaborative study, 22 participant laboratories analyzed our whole cell reference reagent for microbiome from 14 countries. So what the hope is, once we've done a global collaborative study, is that these will be used by all AMR sectors, including industry, government, academia, and clinical labs. So we are currently actively seeking participant labs for our AMR reference reagent collaborative study. If you are interested in being a participant lab, or if you know of other labs that could potentially be interested in this global effort, these email addresses will appear on the final slide, but please come and find me as well if you would like to be considered for this. Just as a side note, I've been told to say that you will be co-authors on any resulting publications as well. So aside from developing reference reagents, we are also interested in how these are implemented in the real life situations. So we actually recently got a publication on the use of our microbiome reference reagents in clinical settings, so in an NHS laboratory, and these actually were involved in improving patient outcomes for patients who had culture negative infections. So the manuscript is definitely worth a look if you are interested. And we have also reviewed the limitations of current clinical AMR detection methods. And there's a manuscript that came out last year, if you are interested. And we have very recently submitted a review paper on the practical steps that you can take for considering reliable and accurate AMR gene detection, as well as considering the key knowledge gaps that are present, which include capturing human and environmental influences of AMR, as well as other complex behaviors like biofilms and tolerance. I'd like to thank everyone listed from the MHRA and Cardiff University who were involved in this effort and the ongoing effort. Without them, it would not have been possible. And these are the email addresses. If you do want to contact me, I'll be around all afternoon. Thank you very much, and I'm happy to take any questions.